I want to talk to you about a test that nearly everyone on the planet is familiar with, the PCR test. Even my 100-year-old grandmother knows what the PCR test for COVID is. And the truth is, we don't really know how it works. And I want to build on the remarks that Professor Alberts had about DNA polymerase and show you our very latest thinking and how DNA polymerase works in the PCR test. Okay, before I get too far into this, I need to disclose that I started a company called Debut, which has licensed the technology at the very end of today's talk. Today's talk will have two parts, and I have to disclose this because if the company succeeds, I will stand to make some money. My university, though, has a conflict of interest policy in place. So there's other faculty, for example, that check my data before I present it to you, just to make sure I'm not overhyping the company. Okay, so today I have two things I want to talk to you about. First, building on what Professor Alberts talked about, I want to talk to you about the smallest units of biology, single molecules that make atoms and bonds, and I want to use this to understand this incredible machine, DNA polymerase, specifically the DNA polymerase that makes the COVID PCR test possible. And then I thought I would switch gears towards the end and show you how young academies can also enhance the science of their members. Because I'm going to show you how through meetings with scientists around the world, I was able to set up a collaboration that eventually resulted in, this idea of using insight from single molecule studies to do stuff, to do organic chemistry at a really massive scale, at commercial scale. So going from tiny, tiny little scale, individual molecules, all the way to monster scale to manufacture chemicals. Okay, so that's the goal of today's talk. And again, that second part was driven through experience that I had as a young academy co-chair over a decade ago. Okay, so first let's talk about PCR. Again, everyone on the planet seems to want to know about PCR these days. And this takes advantage of the incredible properties of DNA polymerase, many of which were highlighted in the last talk. So the way this works is you have some target DNA shown here. And by the way, I'm hoping everyone can see my cursor. You have some target DNA over here, heat the temperature up to 95 degrees. That causes the target DNA to unfold, to fall apart. And then you add these primers that are highly specific for one section of the target DNA. And Taq DNA polymerase, and in fact, all DNA polymerases, require this short section of double-stranded DNA to get to work, to start polymerizing the DNA to make the new strands that were shown towards the end of the movie that Professor Albert showed us. So this is a really effective test because if you don't have a sequence that the primer DNA can recognize, nothing happens. The whole thing is totally blank. So if DNA amplifies, that tells you that the sequence is present. So for COVID, if the thing amplifies, you know that that patient has COVID. And here's the amplification. You start with one copy of the DNA. After one round, you get two copies, and you just keep doing this again and again and again. And oftentimes we'll go for 30 or 40 rounds of PCR. And doing that makes us super duper specific to the point where you can pull out tiny quantities of DNA from, gallons and gallons of sewage, which is really amazing. Okay, so now let's talk about the enzyme that makes this amazing test possible. That enzyme is a thermal stable DNA polymerase called Taq, or Taq DNA polymerase. And, I love the movie that we saw, but movies like that one and this one here, where I'm just showing two structures of Taq running with each other, these movies miss all of the essential detail that we get as chemists. The things I really want to know as a chemist is, when are the atoms and bonds being made? How long does it take those? And most of all, why does Taq work better at higher temperatures? Why do you have to go through this whole process? Why can't you just have the thing working and accelerating the catalysis at room temperature? Understanding that process is difficult. Conventional technologies do not do well at elevated temperatures. The Taq works again at 95 and 72 degrees, and most methods like fluorescence start to fail at those kind of temperatures. The other thing is you really have to get down to single molecule if you want to get to these mechanistic studies, because if you have multiple molecules, oh, and by the way, my cat says hello. If you have multiple molecules, all the details about when the atoms and bonds are formed all get blurred out by having multiple molecules. Okay, so that's our goal for today, is using understanding attack at this atoms and bonds level. So we can do this if we can get the one molecule, the one catalyst, the one enzyme away from all of its neighbors. Doing that allows us to see what I like to call the dark matter of organic chemistry, of chemistry and biology. These are the conformational steps. What is the enzyme actually doing when it's opening and closing? You can see intermediates. Intermediates you never get to see in conventional biochemistry experiments. And also you get at the kinetics and dynamics, which is most of what I'm going to be talking about today. What are the kinetics and dynamics of this bond forming reaction? Okay, so to do this experiment, we, together with a collaborator named Phil Collins, a physicist at UC Irvine, we set up a tiny, tiny little microphone that I like to call a nanophone, because the microphone consists of a nanometer scale wire called a carbon nanotube. And we turn that carbon nanotube into field effects transistor, which is more or less the same as the microphone in my cell phone. Okay, it's more or less the same sort of principle. And then we attach one enzyme to this nanotube using basically a low concentration of enzymes. So on average, you get one attachment. Of course, we have to cover up the electrodes because all biology takes place in salty water, and salty water and electronics do not work well together. So we cover everything up. Okay, so here's what it actually looks like. Here's our exposed carbon nanotube over here, and then here's everything else covered up. And so controlling the concentration of the enzyme and the size of the windowed region, the exposed region, we can get down to one and only one attachment. Okay, so here's one of our first experiments. This is the enzyme that's kind of a workhorse of biochemistry. It's one of the very first ones that we teach our students. This is an enzyme called lysozyme, and it's been known, it was actually discovered by the great biochemist Alexander Fleming. And it's responsible, it's in your tears and on your skin, and it's responsible for bursting the cell walls of bacteria that it encounters. So it's sort of the first line of your immune defenses against microbial colonization. Anyway, so here's the one enzyme attached to the carbon nanotube, shown schematically here, and we have little fragments of cell walls that we're adding. And when an enzyme is all by itself, it's very quiet. Okay, so that quiet is measured by the current on this y-axis. And it's sort of like if you have a speaker, microphones are like the opposite of speakers, but if you have a speaker, the speaker gets louder as more and more current goes through the wire leading to the speaker. So it's very quiet over here. And then we add the substrate and boom, it gets louder. The enzyme, as it's moving, is moving charged residues close to the nanotube, and that generates noise that of course we can't hear with our own ears, but with this very sensitive field effect transistor, we can hear it. So here it is, it's loud, it's chewing on the substrate. And when we zoom in on the details here, so now I'm showing you a third of a second or so. And again, it's same deal, current on the y-axis. Now we can hear the enzyme as it opens and closes. So this is enzyme closed over here, and then enzyme open down here, closed, open, closed, open, closed, open. And each time it's closing, it's breaking a bond that's killing a bacteria. And it does that basically all day long. So it's closed, open, closed, open, closed, open. And no joke, we've done this experiment with enzymes for days upon days. The enzyme kind of shrugs and keeps going. Okay, so speaking of days upon days, we did that actually originally with a DNA polymerase attached to our nanotube. And here's what the experiment looks like. So here's the individual enzyme. We have it anchored non-covalently to the nanotube. So it's sort of like glued to the nanophone. And then we listen in as the enzyme goes about its catalysis. So let's get back to TAC. TAC is this amazing enzyme, operates at high temperatures, has very, very low error rate, something like one error for every 20,000 bases, 20,000 covalent bonds that it makes. And recall that bases have different chemical structures. There's A, C, G, and T, and the A's and C's like to bond to each other, hydrogen bond to each other, and the G's and T's like to hydrogen bond to each other. So that's only one error in getting the correct A with C and G with T for every 20,000 covalent bonds. That's really amazing. Okay, that's truly a remarkable machine. And that's the kind of thing that fascinates me. How does it get it right every time? What are the details and how the bonds are being made? Okay, so to answer this question, we glued different areas of this enzyme to the nanophone, to the tiny little microphone, and each one of those is indicated here with the kind of these purple color blobs. Long story short, and here's what the enzyme looks like attached to the nanotube. This is scanning electron microscopy. That little dot right there, that's our enzyme attached to the nanotube. Okay, so now we start listening in as the enzyme is cranking on its substrate and making covalent bonds. The first thing we find is that the conventional picture of how this enzyme works is incorrect. Okay, the conventional picture really only focuses on these little loops over here. This is from the structures of DNA polymerase that have been solved using X-ray crystallography. And there's just little tiny changes that we see when we look at those changes from structural biology. But when we glue the enzyme in different regions, every single one of those regions undergoes changes upon catalysis. Okay, that's shown here. So this is control. This is enzyme with incorrect substrate. So the substrate is present, but it doesn't allow formation of covalent bonds. And you can see it's totally quiet. Okay, when we add substrate, we see these little blips going downward. Each one of those blips is bond formation. So there's bond formation and there's noise being made as we attach in the intervening region, in the fingers region, in the thumb region. Basically, this enzyme's like a right hand as it closes down to form a covalent bond. In every single one of the regions we looked at, we see the enzyme contracting and collapsing around that substrate to catalyze this covalent bond. Okay, so just looking at structures alone is insufficient for understanding how the enzyme works. You really have to get into the details of how this works. Okay, the next thing, I'm going to skip this. I've already basically said that. Single molecule level tells us something different than we get from these wonderful structure studies that are kind of the basis for this, sort of the foundation of our knowledge in this area. Okay, here's what we learned. First thing we learned is that as you increase the temperature for the enzyme, it doesn't form covalent bonds faster. Okay, that's shown here. I'm showing you, here's room temperature all the way up to 85 degrees. Here is the detailed study of just one of the bond closures, of the enzyme closures where it's forming a bond. Oh, and by the way, we know that is these events correlate with bond formation because the timing matches exactly what's been seen by other people's experiments. Okay, so notice at 22 degrees, it takes about this amount of time. At higher temperature, same amount of time roughly. And this is shown on the right side in a histogram. This is the way my physics colleagues like to show histograms. So this is like 99% of the events over here this is 90% take this long and time and milliseconds on the x-axis, and then 99% take this long. And I'm showing you at different temperatures. You can see at different temperatures, they're more or less the same. The histograms are kind of overlaying with each other. Some small differences, and that actually is kind of interesting, but it's not important for us today. For the most part, bond formation is independent of temperature. As a chemist, this is surprising. I like to think of bond formation as being the prime event in a catalyst life, but actually in this case, that's not the prime event in the catalyst life. Instead, what its prime event really is the eternal hunt for the correct match. Okay, what really gets accelerated at higher temperatures is the enzyme going faster at finding its correct substrate to form the covalent bond. That's pairing the A's and T's and the G's and T's with each other. Or sorry, did I say that right? So sorry, it's A's and T's and then G's and C's with each other. And you can see at higher temperatures, this dramatically changes. The amount of time required dramatically changes. It gets a way less amount of time is required. And you can actually see that here as you go from room temperature. That's the time in between each one of these downward events, these catalytic events. You can see there's more and more catalytic events. The time spent waiting between catalytic events dramatically drops as you go to higher and higher temperature. So that's what's getting accelerated, finding the correct incoming piece, the correct brick delay in the formation of DNA. That's the thing that's going faster and faster. Okay, now the other thing is we found that this enzyme has three states. Most of its time it's spent with its mouth hanging open, waiting for the correct substrate to arrive, for the next brick in the DNA pathway to arrive, the next A or C or G or T. Catalytic events are only 1% of its time or so. But we also see these little tiny blips that are shorter than catalytic events that we call attempts. These are much shorter than catalytic events, but they're taking place in there. There's these tiny, tiny little blips. Oh, and by the way, I need to tell you that the only way we could see these types of events is that we have very, very high time resolution. The time resolution for this experiment is roughly on the order of 1 microsecond. This is much faster than fluorescence experiments, for example. Without that, there's no way we would see this stuff. Okay, so now putting everything together, here is the wild world of Taq DNA polymerase. First of all, it's constantly flickering and doing these catalytic attempts, both when it has matched substrates. This is if you feed it A's, if it has a long line of T's to fill in. So if you have the matched substrates, there's lots and lots of these catalytic attempts. Every so often, though, you get to these events where a bond gets formed. Now, what's really notable is that the enzyme makes these attempts even if it doesn't have a match. So even if it has an A trying to form a hydrogen bond with a C, which we know can't take place, it still closes down. This is sort of like a shopper going through a grocery store. Okay, so it has its shopping cart and it's throwing the wrong items into the shopping cart. It then leans over, closes down over the shopping cart to check against the list. It's checking for the correct hydrogen bonding, but most of the time it's not finding that. And for the mismatched substrates, it then throws out the mismatched substrates and starts the entire process from the start, restarts the entire process. And furthermore, many times it makes those attempts and it doesn't get the correct alignment or something like that. And again, it starts over completely from the beginning each time it does that. So this is like this wild abandon approach of just throwing stuff in, throwing stuff out if it's not correct, and starting from the beginning. Okay, so this idea of thinking about enzymes as molecular machines has inspired me to accelerate catalysts as machines themselves. And this is a Global Young Academy story. Okay, so the Global Young Academy story is I had this idea that enzymes are machines and that they're mechanical machines that have mechanical mechanisms in operation. And I was visiting Australia on GYA business, on Global Young Academy business, and I met this scientist named Colin Raston. He's currently at Flinders University. But we met and he was telling me about this crazy machine that he invented called a vortex fluidic device that inputs mechanical energy into solutions. And again, I'm thinking about enzymes as mechanical machines. And I wondered, can I use this to drive proteins, to drive enzymes, to make them work better using mechanical energy? So Colin and I talked for about 10 minutes. We came up with this collaboration. He sent someone to the United States with one of these vortex fluidic devices in his briefcase, in his suitcase. And we got it through customs, et cetera. And he plugs it into my lab. And almost immediately we find that actually, yeah, proteins can be accelerated very, very rapidly using this approach. And so we've used this for protein refolding. This became this experiment called unboiling an egg that is probably my most famous experiment using mechanical energy to drive protein folding. So that's what I want to talk to you about first. But again, I credit the GYA for this experiment because otherwise I never would have met Colin. And I think it illustrates one of the functions of a young academy, which is to improve the mixing of young scientists around the world and young scientists with each other to establish collaborations that are cross-disciplinary, because you're going to meet people that aren't in your area. I wasn't meeting someone who's looking at mechanical energy. And at this time, Colin was using this for carbon nanotube experiments, for macromolecular experiments, things that I wasn't at all talking to the group of scientists about. So again, that's one of the important functions of a young academy like yours. Okay, so let's get back to unboiling an egg. So I'm going to just show you a video of this because that's the easiest way to illustrate. We're going to start with hard-boiled eggs, And then we're going to unboil them in this vortex fluidic device. And I'll explain how the vortex fluidic device inputs mechanical energy in a moment. And then, because I can, I'm going to cook the egg again. Okay, so we're going to go from raw egg to boiled egg to unboiled egg using the vortex fluidic device. And then I'm going to cook it again. What's happening here is the proteins in the egg, including lysozyme, eggs have high concentration of lysozyme, are going from a folded state in the raw protein to unfolded when you boil the protein. That causes the protein to cross-link and forming that opaque color. And then we're going to use the vortex fluidic device to fold it again. And then I'm going to unfold it when I cook it again. Okay, so that's the process that's going on here. Okay, let's see, let me just turn down the music. Okay, so here's the raw egg. We're cooking it 95 degrees, it's solid. Again, it's opaque. Disaggregate this in 8 mole urea to cause the chains to be in solution. Here's the vortex fluidic device. This is the mechanical energy. It's driving the proteins to fold. And then finally, we're cooking again, okay, because I can. You can actually see the egg over here caramelizing. So it's getting cooked again. Okay, and actually, once you caramelize it, you've undergone a Maillard reaction. So at that point, it's completely irreversible. All right, so what's happening here? There's some transition state energy to go between a folded protein and an unfolded protein. And as you know, temperature doesn't allow you to overcome that transition. When you have a boiled egg, it stays boiled. Even if you heat it up, even if you cool it down in your refrigerator, it's boiled. What we're doing is we're using mechanical energy to overcome that transition state. And that mechanical energy doesn't involve heat. Okay, what's happening is we're using massaging fingers that are operating on the nanometer and subnanometer scales that these mechanical objects of proteins operate at. Specifically, we're taking advantage of shear. So here's the sidewall of the vortex fluidic device. You may recall it's just a spinning tube. And so what's happening is the protein is being, the aqueous solution of protein, the water solution of protein is being forced up the sidewalls of the tube. And then as gravity causes the protein to slip back down, the protein in solution is experiencing shear. It's getting pulled apart. That's these layers in here. In addition, there's these standing waves called Faraday waves. Those Faraday waves are the massaging fingers that operate on these subnanometer scales and cause transition state collapse that cause protein folding and hydrophobic collapse to drive the protein to fold more quickly. Additionally, we've been using this again, thinking about proteins as mechanical objects to drive the catalysis of proteins. Okay, so now I'm showing you the activity acceleration, the catalytic, catalysis of different enzymes, but now accelerated by the vortex fluidic device versus a control left at room temperature on a bench. And these are 2 different enzymes in blue and red. And you can see they have different peaks and valleys of acceleration. You can even notice that these are spaced apart from each other, almost like resonant nodes from each other. Again, we're tapping into enzymes as mechanical objects. And what I'm finding is that there's mechanical resonance within these enzymes, kind of like when you strike a bell with a hammer. The bell hits the hammer, it has particular nodes of resonant sound that it gives off. And another example of mechanical resonance are hummingbirds' wings that flap with this mechanical resonance. So this technology has been licensed to Debut, the company, and they're interested in taking enzymes that otherwise are kind of dogs of biology, enzymes that are found in plants, for example. Plants aren't in particular rushes to produce natural products, but if we can accelerate those enzymes using technology like this, we can drive them to make things in a greener, less energy intensive way with less waste. Okay, so what I've been talking today is engineering and dissecting proteins, thinking of proteins as mechanical devices. I've shown you that there are wild gyrations in the movements of these enzymes during catalysis. And really, we don't see those. We don't see those in most of our experiments as biochemists. And this is something that I'm absolutely fascinated by. For example, the entire TAC enzyme collapses around its substrate. And then the final thing I've been showing you is that thinking about proteins as mechanical objects opens new frontiers for controlling and accelerating those enzymes. And I think that's a really exciting thing to think about. Okay, I need to thank the team that made this work possible. I'm extraordinarily lucky. I work with these amazing young scientists who are shown here. I've already acknowledged the collaborators, and I'm also very grateful to these organizations over here that funded all this research. So thanks so much for this invitation to speak to you today. I deeply regret not being there in person. I look forward to meeting all the members of the Spanish Young Academy in due course. And I look forward to helping you advance your science and helping this young academy reach its highest potential. Thanks so much again for this invitation.